Okay, tools of molecular genetics. Now, this is actually was in the pre-matriculation material. However, I heard most people didn't listen to that. So, just a little bit of background. Genetic testing is used a lot in medicine, and it's used more and more. Genetic testing helps us to understand health issues. It can also allow for treatment of targeted, for genetic symptoms. So understanding genetic changes is going to be important in specific managements, and we're going to talk about that a lot in our cystic fibrosis case. But let's just start back at the beginning. So DNA structure, and you've probably known about this since high school, but I'm just going to review it. We have 4 base pairs, A, T, G, C, adenine, thymine, guanine, and cytosine. The purines are adenine and guanine, and the pyrimidines are thymine and cytosine. Adenine and thymine bind together in a double bond, and guanine and cytosine have a triple bond. And areas of our DNA that have a lot of genes in it, only about probably 10% of our DNA actually carries genes in it. And the GC-rich areas are more gene-rich. So mutation occurs when there's a change in the base. So if it's supposed to be a G, but it switches to an A, so instead of the other band being a C, it's gonna be a T. So depending on where this is, this may or may not make a big deal. Now, this is just a representation of when I'm trying to explain to families about genetic testing. If you think about a chromosome as an encyclopedia, or I like to think about it like a recipe book, we have 23 chromosomes, we have 22 autosomes and the sex chromosome, so we have 2 copies of all of our recipe books. And when we open up a page, that's actually a recipe that tells us how to make whatever the protein is. So let's say we have a sentence, the car was red, and let's say the next sentence is, I drove to the store. What a missense mutation is when there's a change of a letter or a word. So it says the car was hat. So the car was hat, I drove to the store. If I asked you what you did, you'd say I got in the car and I went to the store. If I asked you what color the car was, you'd have a hard time answering that. But if it was the car was RDD, I went to the store, you'd probably even guess that it was red. Just guess. You'd probably just say, oh, it's probably red. Nonsense mutations are when there's premature stop codons, which I'm going to go over in the next slide. And so the car, and that's it. There's no next sentence. There's no nothing. So if I said, what did you do? I got in the car. Where'd you go? I don't know. What color was the car? I don't know. And so these are always disease-causing or pathogenic. Whenever you have a nonsense mutation, it's always bad because there's a premature stop of the protein. Insertions of either a word or a letter can cause a lot of problems. The word, it can change the sequence of the amino acids. Actually, the letter is a little bit worse than the word because it can change every single amino acid, not just add an extra one or delete an extra one. So this is something you've always also probably seen long ago, and this is when we get, so the DNA makes a copy of the RNA, and the RNA is the sequence to the DNA, except for instead of T, we use U. And so if the sequence is UUU, we're going to put in a phenylalanine. There are, like, for instance, in leucine, it could be UUA, UUG, CUU. All these 6 could all put in leucine. So, for instance, if I went from CUC to CUA, I'd still get a leucine. So this is redundancy. Every single amino acid, or protein, starts with AUG, which puts in a methionine. But the important things are the stop codons. So UAA, UAG, and UGA all cause a premature stop codon, and that stops the protein. And that's when the sentence stops. So when I was showing that previous slide. So if we were supposed to be UAC and it went to UAA, it'd go from putting in a tyrosine to a stop codon. versus, as I said, CUU to CUC, we get a leucine or a leucine. So here's another diagram of this. So here at DNA, it's TTC that puts in an AAG that puts in a lysine. But what if we go from TTC to TTT? So we go from a C to a T, we still put in a lysine. This is known as silent. When we're doing genetic testing, this doesn't even make it onto the report because nobody cares. But what if we went from TTC to ATC? Now we have a stop codon, a nonsense mutation. Or if we go from TTC to TCC, instead of lysine, we get arginine. That's a different amino acid, but the good news is they're both basic. But what if we go from TTC to TGC, so instead of lysine, we get threonine? That is actually a polar amino acid, and that could actually be a bigger deal than this one. Both of these are missense mutations. This is conservative because they're both basic. This is non-conserved because it's a different polarity. And then when we're trying to determine if this would actually cause a problem, we have to determine where in the protein it was. So when we make our RNA, it goes to the ribosomes. And here we have our RNA, which was copied from the DNA. And as I said, every single protein starts with AUG, so we put in our methionine. And then we put in our next base and our next base, and now we have our little nice chain of DNA until we get to our stop codon, which releases this protein. The next thing is the proteins go to be folded. And in this case, they usually go into the Golgi to be folded in the correct spot, and then they're brought to wherever they need to be. And here's an example of hemoglobin. So if we had a change of a base that it's at the end here, it may not cause a big deal, but if it's right here where the iron fits in, it could be a big deal. So here's just another representation. We go from GCA, which puts in alanine, to CCA, which puts in proline. That is a missense mutation. But if we go from AAG to TAG, we get a premature stop codon, which is a nonsense mutation. Nonsense is always bad. Just to add some more spice to the whole DNA and how we make more out of what we have, when we go from the copying the DNA, what we have to do is take out the introns. And the introns are non-coding regions. We don't exactly know what introns do right now. Someday we will. But we can actually splice out and this occurs in the nucleus, excuse me. So we could go 1, 2, 3 or 1, 3, 4. And there are a number of genes that when it goes from a infantile, like a embryonic protein to a more mature protein, they have different splicing. And if there was a problem between the intron and exon 4, we might not have an issue in the infantile form, but then we go to the mature form and all of a sudden it doesn't work. So this actually allows us to get, let's say, more bang from our buck from our DNA. The next thing I want to do is talk about blotting and molecular techniques that we use to try to look at this DNA. So the southern blot, we look at DNA. A northern blot, we look at RNA. A western blot, we look at protein. And there's something called a southwestern blot that we use DNA to look at the protein interaction. Disadvantages of blotting is that they don't really amplify signals, so we need like a lot of DNA, like 10 cc's of blood, or RNA or protein. It's very time consuming. And we try to use not as many radioisotopes, but originally we used a lot of radioisotopes. So let's look at mutation screening. So for sickle cell anemia, if only life was as easy as sickle cell anemia. When I went to medical school, the only gene they really knew was sickle cell anemia. And everybody with sickle cell anemia goes from a CAG to a CTG, which goes from glutamine to valine. Every single person with sickle cell anemia has the same base point change. So how do we determine that with molecular testing? One is we use restriction enzymes. There's over 3,500 restriction enzymes, and what they do is they cut the DNA at a specific point, and then we can separate them on a gel and see how big the fragment is. So restriction fragment length polymorphism, we have this restriction enzyme cuts here and here to get our little fragment, but if you have individual 1, it also cuts this third place, so we'll get 2 fragments, but in individual 2, it only cuts like at the ends. And then we use a probe that kind of spans this entire area. So when we add the probe, it will either pick up this fragment or this fragment because the probe overlaps both of them. But in this individual, there's just 1 fragment. So when we separate them based on weight, this is a larger fragment, so it doesn't migrate as far. Or excuse me, from the top to bottom, it migrates. It doesn't migrate as far because it's going this way. But these 2 fragments migrate further, and then we add our probe, and we can see 2 fragments from individual 1, but only 1 fragment from individual 2. Southern blotting. So the first thing we want to do is we cut up our DNA with our restriction enzymes. We separate the fragments on DNA. We then transfer it to a gel, add our fluorescent tag or radioactive isotopes, and then we look at it. When we use radioisotopes, we'd have to use X-ray film, and now we have other methods. But basically, southern blotting is we're cutting DNA, we're separating DNA, and we're determining where the fragments are. Northern blotting is actually looking at RNA. And RNA is how much of the transcript that we get. And so there are actually a number of uses for this in cancer to see, just because the gene is coded, it might be making more protein product than we would like. And so that's something we can look at. And then Western blotting is looking at the protein. And this was used a lot more when there was so many mutations to certain genes that we weren't very good at being able to determine what it was. So we actually looked at the protein got made. So this is kind of the end product. Did we make a protein? We will be talking about Duchenne's muscular dystrophy later in the spring. But dystrophin is the protein. And this is what normal dystrophin looks like. We get a nice band here. Becker's is a milder form. There's actually, it's a shorter protein than it should be. So you can see this band comes a little bit, it doesn't migrate as far. And then with Duchenne's, we get, no protein. So this is normal. This is Becker's. We get absolutely no protein. And that's Duchenne's. So let's talk about, we were talking about sickle cell anemia before, and again, the advantage of every single person with sickle cell anemia has the same change in the base point. So we have a restriction enzyme. In this case, we use MST2. Again, it's not important to remember these restriction enzymes, but it'll cut on either side, and in normal hemoglobin, it'll since this is CAG, so it cuts here, here, and also here. So normal hemoglobin, we get 2 fragments. But when we have sickle cell, it goes from CAG to CTG, so it doesn't cut here. So we only get 1 larger fragment. So this individual has a large fragment and the 2 smaller fragments, so they have a sickle hemoglobin allele and the 2 smaller fragments. This individual, same thing. Their children, the first one, got normal hemoglobin from mom and dad, and so, because all they have is a normal hemoglobin. This individual got the sickle allele from mom and dad, and all they have is a sickle. And this individual has sickle cell anemia. And this individual, like their parents, one parent gave them the sickle allele, and one parent gave them the normal hemoglobin. So this is called. This is the way using restriction enzymes we could figure this out. The next thing is polymerase chain reaction. Again, the problem with using southern blotting or any kind of blotting is requires a lot of material. The great thing about PCR is you can use a very small amount of material and amplify it. So we can have small stretches of DNA that we can amplify with primers. And the way we do this is we cut our DNA, we have our primer, and we amplify it, and we can get a great amount of DNA in a short period of time. So here we have our DNA sequences, and we add our primers, and we heat and cool and heat and cool, and sorry for the ambulance. And then we end up in a very short period of time with a lot of DNA that we can much more easily analyze. So, for instance, Illinois, every state does different things on newborn screening, but Illinois actually looks for sickle cell anemia on newborn screening. And again, the advantage of sickle cell is because everything's the same, we can use a PCR method. So we use two primers. One is for normal hemoglobin and one is for sickle. And then we see if DNA got amplified. So in this individual, all we have is normal hemoglobin. In this individual, all we have is sickle. Okay? This individual didn't work at all. But anyway, so this individual has normal and sickle. So this would be a carrier, normal, sickle, carrier. So we can determine on this ASO test, which is, again, a combination of PCR, if they're normal or sickle or even carrier. The next thing I want to talk about is DNA sequencing. What is the code of the DNA and how do we know if it's normal or abnormal? So the first thing we needed to do is sequence all the DNA. Sanger sequencing is the gold standard even today to sequence a gene. What we do is we have a solution. We use our 4 normal bases, ATGC, and we also have deoxyanalogues. And when the deoxyanalogue is added, the chain no longer extends. So let me just show you this. So we have our little fragment of DNA, and I would just like to say a lot of people got their PhDs doing Sanger sequencing, picking a gene, and sequencing it. This is a very tedious, expensive, time-consuming, but incredibly accurate method to sequence DNA. So we have our fragment of DNA, and in this solution, we add our fragment and we add ATGC and our deoxy-T, and whenever deoxy-T is added, the chain terminates. And the next, solution, we have our AGTC with our template, and we have a deoxy-C, here again, deoxy-A and deoxy-G. So, in this bottom here, as we're trying to sequence our DNA, GACAG, and then it puts the deoxy-T right away. So we get a very short fragment. And this one, the first time it put the normal T, but the second time deoxy, and then this way it made all the way to the end. And so we have 3 fragments that are different sizes, so on and so forth. And from this, we can then, we run all these on a gel simultaneously, and then we T, A, T, C, C, A, G, T. So this is a little video on this. And if I turn the sound on, it will actually work. Here, let me turn the sound on my computer.
Must first be obtained in single-stranded form. A mixture containing the single-stranded DNA, DNA polymerase, the 4 deoxyribonucleotides A, T, G, and C, and a single short primer is prepared. The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. Equal amounts of this reaction mixture are placed in each of 4 tubes, and a different dideoxyribonucleotide is added to each tube. When a deoxyribonucleotide is inserted into the growing chain, replication continues. However, when a dideoxyribonucleotide is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and nucleotide type. The shortest oligonucleotide moves furthest down the gel. Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.
Oh. Okay, I don't know what happened to this. Huh. Anyway, we'll just move on and stop talking. So, reading DNA gel sequence, again, we can see what the codes are. And you can see this is incredibly tedious and time consuming, but the gold standard. So let's talk about cystic fibrosis, which is the case. Unlike sickle cell anemia, which was the easy thing, there are over 1,400 mutations we know of in CFTR. Some only occur in single families. These include missense mutations, frame shift, splicing, and nonsense mutations. But delta F508 is the most common. Over 70% of patients with cystic fibrosis, at least one of the 2 alleles, because cystic fibrosis is inherited in a recessive fashion, which means there's 2 mutations, have at least one is a delta F508. Now, one thing I want to talk about is mutation screening. So if we're looking for common mutations, and there's certain ethnic groups that they're really common in, then let's just, in the Ashkenazi Jewish population, 94% of individuals will have a detection of a CF mutation looking at common mutations. So the risk of having cystic fibrosis before genetic testing is really high, 1 in 24. However, if it's negative, because there's a 94% detection rate with common mutations, it goes down to 1 in 400. Caucasians, we have an 88% detection rate. And the prior risk is similar to that of the Ashkenazi population, 1 in 25, but because going from 94% to 88, our after testing is 1 in 208. Hispanics, although, only have 72% detection rate. So even though the risk is much lower than Caucasians, their risk is still, and it's still a little bit lower afterwards, because we're only picking up 72%. African Americans, again, even lower, about 1/3 of that of Caucasians, but we only pick up 65%. And then Asian Americans, which it's almost 1 in 100, 1 in 94 before you take the test, but there's only 49% detection rate. You're not really dropping the risk all that much in the post, risk after genetic testing. And that's kind of an important concept. It's a difficult concept, but if you're picking up most of the mutations, then you go from the highest risk to really the lowest risk. So for cystic fibrosis, there are panels that look at the common mutations, and this is very cost effective. However, negative testing does not rule out being a carrier, especially if you're of Hispanic or African American or Asian descent. You haven't really decreased your risk all that much because these are rare mutations. Mutation screening benefit versus sequencing is it's cheaper, it's quicker, it looks at common mutations, so you can do it as far as population screening. It's a good method. Limitations, it doesn't tell you about rare mutations. So the way that we do this for prenatal carrier testing is we will test the mother for common mutations. And we all carry genes that don't function correctly. We all carry about 6 to 8 genes that don't function correctly. And so we'll test mom for these rare recessive disorders. And if she's a carrier of something, we then test dad. But when we're testing dad, we don't want to just do carrier screening for that gene. We actually want to sequence that gene to make sure 100% he's not a carrier. So here we have a USMLE question. A researcher is trying to, and I took this out of a book, a researcher is trying to identify a specific protein in a mixture. He subjects the mixture to gel electrophoresis and transfers to nitrocellulose filters. The filter is incubated with antibodies to a protein. The excess antibody is washed off, and the protein is then incubated with a radioactive protein that binds to the antibody. Autoradiography is performed for the presence of the protein. Which of the following is this? So, he's trying to identify a protein in the mixture, and he transfers it, and he incubates it, and he uses an antibody with a protein to figure out where it is. So we're talking about protein, so this is a Western blot. A Southwestern blot would be if he separated the protein and used DNA against it. So, which I swear to God, I've never even heard of a Southwest blotting until I was reading like some of these USMLE questions. I had to actually look it up. Anyway, so this is Western blotting. And that concludes this